Hello guys, welcome to Doctor Science. So today we are going to talk about the immediate versus late phase reactions seen in type 1 hypersensitivity reaction. So in this we will be mainly talking about the products of immediate and late phase reactions. So let's start. So this is a mast cell, and this mast cell is having these receptors. What are these receptors? These are Fc epsilon Ri receptors. Okay. So these receptors are activated by IgE antibody, and once this IgE antibody are cross-linked, the mast cells are activated, right? And these mast cells, they have these vesicles inside them. In these vesicles there are granules. These are the granules that are present in the mast cells. So mast cells are activated and these granules are released outside, and they will cause the inflammation. So this can be divided into two phases. Okay, one is the immediate phase. Immediate phase means it happens less than 2 hours. And the late phase reactions, late phase means it is greater than 2 hours, 2 hours to 24 hours. Okay, in this immediate phase reaction, let me clear it, you will be seeing mainly two types of granules. Okay, one which are lipid derived and the cytokines. Okay, and the other one is the preformed mediators. So first we will talk about preformed mediators because these are very important. What is meant by preformed mediators? We have to know the definition of preformed mediators. So suppose here is the mast cell. They have these vesicles. In these vesicles, these are the granules present. Okay, so it means this mast cell is storing these granules in a vesicle, and it is keeping it aside. Okay, it means it is preformed, whereas lipid derivatives and cytokines, they are spontaneously produced. Once the mast cells are activated, they are spontaneously produced by de novo synthesis. Okay, whereas these preformed are stored in vesicles and they are kept for future use. Okay, so once the mast cells are activated and these preformed mediators, what are the preformed mediators? We will see. The first and the most important one is the vasoactive amine. Vasoactive amine. What are these vasoactive amine? These are the active amines. These are the amines which will activate the vessels. Okay, hence they are called as vasoactive amine. These are histamine. We can call it as histamine because it contains amine group. Okay, so what does the histamine do? This is the most important thing to know, because in immediate type, immediate phase, which is less than 2 hours, we will be mainly seeing histamine. Okay, in the late type we will be seeing mainly we can say prostaglandins and all other things, but we will discuss this one later. So in this immediate phase, in the preformed vesicles, we can say the histamine is most important. And this histamine, what it does, it increases the vascular permeability. What is meant by vascular permeability? So this is a blood vessel here. So what happens means, these endothelial cells present here, they are tightly packed. So this histamine and goes acts between these endothelial cells, and what happens? So here is another blood vessel which is acted upon by histamine, and these endothelial cells, they will allow this fluid to leak out. Okay, so this is called as vascular permeability. It is nothing but the fluid will leak out. And what happens when the fluid is leaked out? it causes edema, right? So histamine it causes, the first one is, yeah, increased in vascular permeability, which will cause edema, and the other one is it also causes bronchial smooth muscle contraction. What is meant by smooth muscle contraction? So here are the bronchus and here are the alveoli, and here are the smooth muscles. Wait a second. So here are the smooth muscles, and these smooth muscles will contract, and once they are contracted, this bronchus is like uh, the air cannot go inside, so there is decreased breathing. Okay, I mean decreased oxygen going to the lungs, right? So what is histamine doing? The first one was increased in vascular permeability, which will eventually cause edema, and the second one is increased in smooth muscle contraction, which will cause breathlessness or decreased oxygen going to the lungs. Not only that, there are these cells. Okay, so here are the cells, and in between these cells there is goblet cells. These goblet cells, they have receptors for histamine, and once the histamine goes and binds with them, it will increase the mucus production, and this mucus is not good. Okay, this will obstruct everything, so it increases the mucus production also. Histamine will increase the mucus production mainly in the intestines. So these goblet cells are present in the intestine, and you can say in the nasal area, and okay, you can, and in the lungs. Okay, so those are the three functions of histamine. Next we will see what, so we talked about histamine earlier. What are the preformed antibodies? So under preformed antibodies, which means they are already formed and stored in vesicles, histamine is over. Next we will talk about chimases, tryptase, and acid hydrolases. All these three, they will cause tissue damage. Okay, so we talked about histamine, chimase, tryptase, and acid hydrolases. And what is the third one? The third ones include heparin sulfate, or chondrin sulfate. Heparin sulfate, or we can call it as heparin. Okay, this heparin is an anticoagulant. So suppose this is a mast cell. Wait a second. So this is a mast cell, and this mast cell contains mainly preformed antibodies. We will talk, preformed. Wait a second. Okay, so here is the mast cell, and we will talk about what are the preformed mediators of this mast cell. So it contains histamine, right? The first one is histamine. So what are the other things? Chimase, tryptase, and acid hydrolase. And what is the last one? Yeah, heparin and chondrin sulfate. Okay, so these are stored together. So these were the preformed mediators of mast cells. Now we will talk about the lipid derivatives, lipid mediators, and cytokines. So what are these lipid mediators and cytokines? Okay, this lipid mediators, so suppose this is a mast cell. I will take a part of this mast cell and I will draw it here. This is the membrane of the mast cell. Okay, This is the phospholipid membrane of the mast cell. So there is an enzyme. Okay, there is an enzyme. Here is an enzyme which will convert these phospholipids. These are the phospholipids, right? So they will convert these phospholipids into their desired components. Okay, and the enzyme here which is catalyzing these phospholipids are called as phospholipase A2. So this phospholipid A2 will convert those phospholipid into mainly leukotrienes and prostaglandins. Okay, so we will talk about the main prostaglandins here because these are very important. In this prostaglandin we will talk mainly about B4, C4 and D4. Okay, wait a second, this is not C4 and D4, it is C2, prostaglandin C2 and prostaglandin D2 and prostaglandin B4. So what does this prostaglandins do? They have this work, right, to do. So we will talk about prostaglandins C2 and D2. Okay, let's talk about what are the functions of prostaglandin C2 and D2. So prostaglandin D2, C2 and D2, they are the most potent vasoactive. As I already told you what are the function of what is meant by vasoactive, and spasmogenic. It means it will cause smooth muscle contraction. So they are the most potent vasoactive and spasmogenic agents compared to histamine. So if I give a quiz about prostaglandin C2 and D2, which of the following are vasoactive and spasmogenic? So it will always be prostaglandin C2 and D2 compared to histamine. Okay, compared to histamine, prostaglandin C2 and D2 are more vasoactive and spasmogenic. Okay, you have to remember that. Next we will talk about here this is prostaglandin, that is we will talk about prostaglandins C4. Wait a second. It is prostaglandin C2 and D2. Okay, leukotriene B4s are different thing. I just got confused there. Okay, so next we will talk about leukotrienes. In this we will talk about leukotriene B4. What does the leukotriene B4 do? It will attract more neutrophils to the site of inflammation. So that is the function of leukotriene B4. Okay, so that was about the lipid derivatives. Next we will talk about cytokines. In the cytokines we will be mainly seeing tumor necrotic factor and interleukin 1. So tumor necrotic factor and interleukin 1, they will attract leukocytes. Okay, so let's talk about what are the contents of immediate phase. Immediate phase is less than 2 hours, and in this we can see mainly preformed mediators. Preformed mediators are histamine, chymase, tryptase and acid hydrolysis, and the last one was heparin and chondron sulfate. Okay, and lipid derivatives are leukotriene B4, prostaglandin C2 and prostaglandin D2. There is also platelet activating factor, but it is not very important. Okay, so these were the cytokines. Okay, in the cytokines we will see mainly tumor necrotic factor and interleukin 1. What is the function of these? They will attract leukocytes. In this, leukotriene B4 will attract leukocytes. Okay, so that was about immediate. Next we will talk about late phase reaction. In this late phase, it happens between greater than 2 hours, 2 to 24 hours, and it happens in the absence of stimuli. It means again a antigen should not come and bind, and all that shit is not needed. Okay. In this late phase, we will be mainly seeing eosinophils. Okay. Since it is mainly T helper 2, T helper 2 releases interleukin 5. Interleukin 5 is the most potent activator of eosinophils, and we will seeing mainly eosinophils in late phase reaction. Okay. So that was about immediate and late phase reaction. So what are